Hello, I'm Ouyang Teng, and with me is Larry Hecht, editor of 21st Century Science and Technology magazine, who just wrote an article entitled New Evidence for a Non-Particle View of Life. The prevailing view in science today, sometimes known as reductionism, is that life results from the combination of non-living molecules. Get together the right mix of amino acids and other basic ingredients, put them under the right conditions, and they will spontaneously come together into a primitive living organism, from which more developed organisms can evolve. Such a view has never been demonstrated, and indeed every attempt to carry out such an experiment has failed. What has been recently presented as the creation of a primitive living form, or a proto-living form such as DNA or a virus, always requires the presence of another living form. No one has ever taken a collection of non-living molecules and turned them into life. An alternative view, known as vitalism, argues that living organisms contain a force, a life principle, distinct from the matter that makes them up. Life is the combination of the right organic molecules with this living force. But no experiment has ever detected such a living force, or demonstrated that it is present within a living organism and absent in the same collection of non-living matter. What is life, then? Today we are going to examine a series of experiments that shed new light on this question, not by answering it, but by raising some provocative questions that will help us to formulate for ourselves what it is we mean by life. The experiments were carried out by Luc Montagnier, the leading French virologist who won the Nobel Prize in 1983 for his discovery of the HIV virus. Among the fascinating results observed by Montagnier and his team were the emission of low-frequency electromagnetic waves from bacterial DNA sequences, and the apparent ability of these waves to organize nucleotides, the raw material of DNA, into new bacterial DNA, by mediation of structures within water. By revisiting the question of the interplay of radiation, including atmospheric and implicitly cosmic radiation, with life, the work has revolutionary implications for biology and our fundamental view of the universe. Before Larry and I get into our discussion of this subject, we're going to present to you an illustrated summary of what Montagnier and his collaborators did in these experiments and what were their results. Dr. Luc Montagnier first noticed the remarkable interaction of bacteria and water 10 years ago while studying a small bacterium known to be a frequent companion of HIV, the Mycoplasma pyrum. Like HIV, this bacterium loves human white blood cells, also known as lymphocytes. Starting with a culture of human lymphocytes infected with the bacteria, Montagnier then passed the culture through filters with pores small enough to prevent the passage of the bacteria and lymphocytes. What passed through the filter was the fluid in which the bacteria had been cultured, minus the bacteria and lymphocytes that had been filtered out. Further tests on this fluid filtrate showed that it was indeed negative for signs of the Mycoplasma pyrum bacteria. Then, a curious thing happened. When the filtrate, containing no bacteria, was incubated with a new batch of bacteria-free human lymphocytes, the original Mycoplasma bacteria was found to fully regenerate within the new lymphocytes. Where had this bacteria come from? The evidence pointed to an exciting possibility, that somehow the original Mycoplasma bacteria had transmitted into the structure of the fluid medium itself the information needed to regenerate itself at a later stage. But that wasn't all. Next came the big surprise. After multiple dilutions with highly purified water, the apparently sterile filtrates were found to produce low-frequency electromagnetic waves, which were detected after the filtrates were placed inside a copper coil attached to an amplifier. Waves in the frequency range of 500 to 3000 Hz, within what is considered the low end of the radio frequency spectrum, were observed in the dilutions of many different kinds of filtrates, in solutions that had been infected with bacteria and viruses, in the plasma of the blood of humans infected by these same agents, and in the diluted filtrates of DNA extracted directly from these bacteria and viruses. One other important condition was required for the emission of such waves: the presence of an electromagnetic background radiation of about 7 Hz, which corresponds to the approximate frequency of the natural resonating cavity formed between the surface of the Earth and its electrically charged ionosphere, known as the Schumann resonance. This frequency could also be produced artificially in the laboratory. In further experimentation, Montagnier showed that different dilutions of the same filtrates could communicate with each other in a form of crosstalk. To test this, a sample of non-emitting dilution was placed alongside a sample of emitting dilution of the same species. The non-emitting sample appeared to silence the emitting sample, indicating that some type of electromagnetic signal does indeed emanate from the seemingly non-emitting sample, but which, perhaps because it is too weak, is not recorded directly by the copper coil. In another strange twist, when the now silenced sample was further diluted, an electromagnetic signal reappeared. We have already seen that species-specific electromagnetic signals emanating from the water of the diluted filtrate seem to be connected to a species-specific imprint in the structure of the water itself. The next phase of experimentation showed this in an even more dramatic way. A fragment of DNA from the HIV virus was used as the source. A solution containing a number of copies of the DNA fragment was filtered and then diluted until an electromagnetic signal was obtained, as in the previous experiments. Then a further filtration and dilution was carried out on both this sample as well as on a sample of pure water. Both samples were then placed inside a copper coil, which was inside a box of mu metal, which has the property of blocking out external low-frequency electromagnetic fields. A low-intensity 7 Hz electric current was fed through the copper coil to generate the stimulating background radiation. After 18 hours, the tube containing pure water was removed and found to emit its own electromagnetic signal. But the most remarkable step came next. The organic ingredients normally used for making copies of DNA were then added to the tube containing pure water. Normally, for these raw materials to be able to assemble multiple copies of DNA, at least one strand of the DNA to be copied needs to be present to serve as a template. In this case, none was added, and the pure water sample, with the added ingredients of nucleotides, primers, and polymerase, was subjected to the normal cycle of heat exposure used in the process of DNA replication. The results were astounding. Not only was a DNA fragment produced within this sample, but it was 98% identical in sequence to the original DNA fragment of HIV. How is this regeneration from water possible? Just as with the pure water sample, in all these experiments with bacteria, viruses, and their DNA, the emitting filtrates are so highly diluted as to have almost no likelihood of containing the original genetic material. To account for the measured effects, Montagnier has adopted a hypothesis developed by a number of researchers into the anomalous properties of water, namely, that in a liquid state, water is not a random agglomeration of molecules, but rather that chains of water molecules can constantly form coherent nanostructures, which may be both the products of, and the sources for, electromagnetic resonance phenomena. While Montagnier's experiments are not yet conclusive, they are nevertheless consistent with a rigorous experimental approach that goes back to such figures as Alexander Gurevich, who had shown in the 1920s that the emission of electromagnetic radiation in the ultraviolet range was involved in the process of cell mitosis. More recent work by Fritz Popp and colleagues has confirmed that the source of this UV spectrum light, which he named biophotons, is the DNA in the cell nucleus, and that biophoton emissions correlate with known biological rhythms of diurnal, lunar, and other periodicity.
There's a shocking element to this, But in another sense,
There are
it
many
is
important
something
questions
which
posed by
harkens
Montanier's
back
work
to
so far.
the kind of experimental
Further
work that we saw
experimentation
with
and further
Gurevich and
refinement
others.
of the instruments involved must obviously be carried
The
out to
shocking
begin to
element is that
answer
it
these
is
questions.
an in-your-face kind of
A broad view of the
demonstration
results
that
of
life
Montagnier, however, touch
is not
upon a most
restricted
fundamental
to
question
the
as to the
presumed
nature of
kind
life.
Simply
The principle
biochemical
first elaborated by Francesco
view,
Redi in the
biochemical
17th century
Yes.
and later
of,
emphasized
sort
by
of,
Vladimir
infinitely,
Vernadsky, that
a
all life
continuing
comes from
complexity
life,
of
has
parts
never been
coming
shown to
together.
be
And
violated
if we
in
can
any
only,
experiment to
get
date.
smaller and smaller and
But in
find
discussing the
those
principle,
fundamental
it is
parts
usual to
that
envision
will
some
eventually be able to put,
material
Humpty
process,
Dumpty back
such as
together
egg
again
and sperm,
and
spore,
get
or
life
cell
in the
division,
large.
as the responsible agent.
And here you've got
In
clearly
the results
something which
reported
is,
here, however,
which is not
the
only,
life principle
uh,
appears to be
something
transmitted,
which
at least
expresses
in
an
part,
interaction.
not by the immediate presence of a
The idea
material
that you have
substance,
Yes.
but
bacterial
immediately
or,
in connection
DNA,
with a signal
bacteria,
detectable
DNA,
as electromagnetic in origin,
but in an
and
intimate
seemingly connected to
association
broader electromagnetic
with
characteristics
that
of the
class that
environment.
supposedly
The further
other,
elaboration of the
alternative
mechanisms,
class of phenomena
whether
that
by
we call
formation of
radiation.
nanostructures
Yes.
in water as
But
suggested,
sort of both are
or
necessary.
perhaps
What
by
you
additional
see
means,
as the
remains
outcome
to
is
be
a kind
worked out. What is clear is that the results require an extension of that usually limited conception of life. The principle of all life from life still holds, but only on the condition that we adopt a non-particle conception of life. So Larry, I mean, when I first read about this, it resonated in one sense because there's, as we mentioned, this is
Resonated, that's a good word. for
right,
this.
exactly. In one sense, there's a shocking element to this, but in another sense, it is something which harkens back to the kind of experimental work that we saw with Gurevich and others. The shocking element is that it is an in-your-face kind of demonstration that life is not restricted to the presumed kind of simply biochemical view, the biochemical construction
Yes.
of sort of an infinitely, a continuing complexity of parts coming together. And if we can only, get smaller and smaller and find those fundamental parts, that will eventually be able to put, Humpty Dumpty back together again and get life in the large. And here you've got clearly something which is not only something which expresses an interaction, right? The idea that you have
Yes.
bacterial or DNA, bacteria, DNA, but in an intimate association with that class, that supposedly other alternative class of phenomena that we call radiation.
Yes.
But sort of both are necessary. What you see as the outcome is a kind of intersection
Yes.
of a process.
And then, of course, here we have a demonstration, assuming we don't have an experimental error somewhere, we've got a demonstration pretty hard to deny of radio frequency, low frequency radiation interaction between bacterial cells, or even the sliced up DNA or DNA pieces of the bacteria. But then we also know of interactions with cosmic radiation, with gamma radiation, with what we call nuclear radiation. All of these interactions have a relationship to life. Sometimes we isolate here in the laboratory a particular thing. This is the whole broad band. And what is radiating? This is something we haven't, probably the work of Pop on which you mentioned in the narration. Here we have a radio. We've got the whole electromagnetic spectrum. There must be something going on everywhere, including sound also. But here we have something in the range of they say the dominant signals around 1,000 hertz, which is that actually, if that were audio, that would just be a high C, and double high C, a little below it. There's a range, he said, he detects between about 500 and 3,000 hertz. Then we have light frequencies way up there, considerably higher, that have been detected. We have known interaction with nuclear radiation, with electrons. All of these things can be looked at as wave phenomena. So we've got a whole array. And what is it doing? What is it doing to the organism? What is the organism doing with it? At a certain point, this separation, that separation in your mind has to break down to really get to the next step on this thing.
The separation between organism and maybe environment.
Yeah,
sort of
the part in the whole.
Right.
We've reduced the part. Yeah, we've reduced the part down to, okay, this living organism, and then you want to take the living organism apart, and then you've got these pieces. You've got the DNA, which, by the way, is just it's a somewhat overemphasized part of the whole deal.
Mm-hmm.
You know,
the emphasis on DNA as
DNA alone is it's an important piece of things, but that's not everything either. But then, it appears in Pop's work with the light radiation, which is the extension of Gurvich's work all the way back in the 20s. You've got you seem to be able to isolate the DNA as the source of the signal, and that is certainly intimated here too, because the signaling occurs even. So, is there something? So, the structure of the DNA, the emphasis is always on the biochemical. A certain amount of geometry considered in the thing, but what else is it doing? Is it just a chemical signaler, or is it? Well, it's putting out light and receiving light. It's putting out low frequency radio and receiving it. Mm-hmm. We don't know the whole of things. We've always tried to. The whole emphasis has been to try and reduce biology and even cognition to the laws of physics and chemistry.
Right.
It's really got to be the other way around. The higher domain defines the lower domain. The laws of physics and chemistry are incomplete.
Well, it's interesting. You point out Gurvich's work and then the extension by Pop on these so-called biophotons, or emission in the ultraviolet range, and then now Montagnier's work showing emission in the radio range. As you point out, you've got implicitly between that the whole spectrum,
yes,
right?
What else is there?
You've got photosynthesis in the visible range, and
right.
it really does give you, as far as saying, well, let's redefine physics from the standpoint of biology, minimally, right? Then you already have a sort of, it seems like the raw material to begin even redefining what we mean by that range of radiation.
Yes.
It's functionally and qualitatively, they have the different radiations have qualitative characteristics which are not expressed just in the abiotic kind of instrumentation that has been used to define them.
Absolutely. I think we'll find these the famous paradoxes, the fascinating wave-particle paradox, these sorts of things that come up. I mean, that's an indication of incompleteness when you get to things like that stick there and they stick for 100 years. There's something missing. You got to go above that, go behind it.
Right. You point
Look
out
around the back and see what's really ticking there.
Right.
You know, and you're going to find your model of the thing is all wrong. Your conception even of what it is, it's something much larger.
I mean, you point that out in the article, in terms of what Schrödinger tried to do.
Yeah.
As a physicist who was really dealing with the wave particle, paradox as it's called, to say, is there a, how would a physicist define life in the context of all of this work that was happening in the early part of the 20th century, that had kind of run up against you might say it really did run up against the limits of abiotic physics, to see that you had on the one hand phenomena that seemed characteristically particulate and corpuscular, and phenomena on the other hand that seemed irreducibly wave and continuous, wave-like and continuous. And as you point out, he wasn't able to resolve that. But because, frankly, he was operating within the wrong phase space.
Right.
He was trying to take life and bring it to what he knew, and what he was
something
competent
that he
in.
helped to define in physics, but got stuck in his own box, huh?
Yeah.
Well, that's that famous paper of 1946, Schrödinger's paper, What is Life? I think it was 46, which is, defines an exciting question, but, it doesn't get out. Well, he gets trapped in this entropy thing. Well, life is obviously not entropic, but he ends up, it's too much of a compromise. He ends up with, well, entropy rules, but here's a local exception to it.
Yeah.
Right, and it's an exception, and that's still what's widely accepted because it's permitted. You know, it's important to get into the history of all of this. One thing to realize, Montagnier having to go to China,
Right.
and he's probably happy to go, but he's from France. He's working in a European environment, and people have such a they have such a reaction to this thing. I've seen it. I've put this out among certain professionals in this field.
The article.
And first, just not even mine, just what he had done, and people just have the heebie-jeebies over the thing. They go wild, they go ballistic. You just raise the question. I had a long dialogue with somebody, and I found out after 10 or 12 emails back and forth, the fellow hadn't even read the original article. He's giving me all these objections to it.
Well, what are their objections? What is it that's so scary?
It's verboten to bring in this question of radiation. Gurevich already faced this. Gurevich went through the Rockefeller Institute, mobilized to shut down any attempt to pursue Gurevich's work. And we had one of our colleagues had an interview, talked to the guy much later, like in the late 80s or early 90s, who had done it, in 1938, who had ruled that Gurevich wasn't to be pursued, and basically admitted then, 40 years later, that there was no 50 years later, that there was no evidence to say what was being said against Gurevich, but it was just ruled out. The same thing happened. There was work being done in America too in this direction. That direction of things was shut down. You weren't to go in the direction because, well, what is the fundamental issue is the fundamental reductionism is a state of mind. It's Aristotelianism. It's the conception, if man is something higher than a bunch of mud, then that's the Republican principle too, isn't it? Are we just a bunch of mud or clay that's been packed together into something? Well, the modern way of saying that is you're reducible to molecules that obey the laws of physics and chemistry. Well, sorry, buddy. The laws of physics and chemistry are going to be defined, and we're going to have fun redefined. And we're going to have fun doing it when we get to a higher understanding of life and the interaction of life with the universe.
The funny thing is they say, yes, life is subject to the laws of physics and chemistry, except where there are exceptions. Right? As you pointed out with the entropy principle, which is to the extent that certain experimental evidence has to be accepted, it'll be accepted as an exception.
Yeah.
And so you sort of have a strange case of a science of exceptions instead of recognizing that the universe is fundamentally negentropic or anti-entropic. And that ought to redefine the way that people then go back and look at the so-called fundamentals of science. So if it's not dismissed outright, then it's considered, well, this is a funny little exception that has to be boxed in. And I think one thing that's interesting, as was mentioned before, is that the effect that Montagnier was studying seemed to be dependent on, so-called external electromagnetic activation or stimulation.
Yes.
Something, which could possibly be related to the Schumann resonances of the surface of the Earth and the ionosphere, which, the ionosphere itself is in intimate connection with solar and cosmic radiation. Montagnier also points out that, a similar frequency range as is produced in this kind of biospheric resonating cavity of the whole Earth is produced in higher organisms like ourselves in our central nervous system. Which sort of gave me the idea of a kind of a nested relationship. On the one hand, you're taking some relationship which may exist with respect to this a microbial organism in the biosphere as a whole, and a sort of evolutionary process is able to encapsulate that, perhaps, in a higher organism. But either way you look at it, it's clear that the question of this broader environmental, so-called environmental aspect to what's happening in the small.
Yes.
It forces you to recognize that really you are dealing with even if you can't account for all the principles at the time, you're dealing with sensitivities to processes which are on a much larger scale.
Right, and then you've got the question even within biology: what is an organism? You know, well, so if you have this communication, which includes this wave-like communication of multiplicity of frequencies, here you're dealing with very simple things, relatively simple things, the bacteria. Then you get organisms that are themselves collectivities, that are like the siphonophores, things that are collections of separate, actually can go off and be separate organisms and then can come together and function as a whole. Then you have us, and of course, you know, you have mammals and then you have human beings, and we're different, but biologically speaking, something that is a collectivity of many, many different kinds of cells, all right, operating together. If those cells are doing this signaling, and this signaling is amongst them and also to the outside, right? And then, of course, there's all our 10-to-1 ratio of commensal bacteria, which is always a fascinating thing too, and they're in there with us.
You mean the fact that we've got 10 times as many bacterial cells in our body than we do of our own cells?
Yeah,
right.
And they're talking to each other too. Then there must be some very interesting signal going on there in this whole collection of things. You know, it really adds a new dimension to what de Broglie and Schrödinger were interested in. The idea of every piece of matter also having a wave-like property. This is a much more complex wave-like property than you can write in that equation.
Yeah, polyphonic probably is something that they hadn't maybe considered at the level that it needs to be.
Yeah, at the very least, it's a Beethoven symphony.
Right. If only Beethoven had gone into biophysics, huh?
Yeah, well, that's what you need.
Yeah. I mean, the other thing that strikes me about the experiments here, you know, there's this question of water. I mean, the mediation through water. In one sense, there's another area which maybe is not so surprising, given the fact that we know that cell cytoplasm, which for a long time, or I think maybe, you know, people still sort of have this implicit idea that the cell is just kind of a bag of water.
Oh no!
But it's very structured, and the fact that, you know, from that we already know that you're dealing with a material substrate here, which is susceptible of a very high level of organization, and it's now being in these experiments in the test tubes, you know, with these diluted filtrates. It seems like you have an echo of that kind of organization, maybe on a slightly larger scale, and now connected to this very distinct emission of low frequency electromagnetic radiation.
What's beautiful about it is the ambiguity of it all. The very thing that people react against. You know, there's this instinctive reaction. There are levels to it. I was speaking before of the. There is an ideological level to this thing of the demand, this insistence on reductionism. It's the Thomas Huxley program within biology. You know, that's been defined. Literally defined within the British Empire since the 19th century, but there are other levels. There are other levels to the thing. There's this natural resistance to people who think they have it all figured out. You know, it's upsetting to them. As people get older, they've mastered a lot of things, and you know they don't want to be challenged any further. You get a lot of that with these kinds of things too. So it's better to stay alive, to use a word. And be attuned to the exciting nature of these new things. This is just fun. I mean, we're just scratching the surface here. We're just defining what we've got here. We're just defining a new problem, and it's got to be worked out. Maybe some of it'll turn out to be error. The whole thing is an error. There may be parts of it may turn out differently. We'll try different things with it. Find out what's going on there. And the whole thing everything begins with something that doesn't quite fit. You know, it began as you pointed out in the narration. It began with this culturing of a bacterial culturing of these mycoplasma from what appeared to be a sterile solution. How did that happen? We don't know. One of my immediate thoughts, since I had been looking in this direction on this interesting, fascinating question of the bacterial pleomorphs, that bacteria were the distinction between bacteria and virus, well before we had electron microscopes, was originally defined as filterable or non-filterable. That is, how small could they get? And there was a fascinating, a famous paper in 1948, I think it was, by Emmy Kleineberger, someone who'd come out of Göttingen and had to leave Germany, ended up at the Lister Institute, who wrote a paper called Filterable Forms of Bacteria. Well, the upshot of this thing was it was a pulling together of work that had been going on for many decades, and then her own follow-up work on it, demonstrating that bacteria go through life cycles. You could prove it, and you could culture them in different stages of these life cycles, from including some that are very small. But it occurred to me, well, perhaps you're reculturing a pleomorphic form that had passed through the filter.
Something that took on a form that allowed it to pass through.
Yeah. Well, there was a control for that. There was this attempt at use these PCR techniques, these DNA amplification techniques.
Mm-hmm.
So we presume, but what are the limitations of that? We don't know. We presume that if the DNA is there, we're going to find it, we're going to culture and amplify it out of the, prove that it's there. But I don't know. Maybe it's hiding in a lymphocyte or something. There are until you take experimental work like this and people do it and other people do it, not from the standpoint, don't give it the treatment they gave cold fusion. It's got to be disproven. So a bunch of people did some phony experiments and said, oh, it didn't work for us. No, take it on as a really fascinating challenge and look at what's going on there. Every step of the way, I think some new things are going to be discovered.
Right. I mean, there is, you mentioned the question of disease. That's one of the things that's obviously tantalizing about this kind of work. Yes. If you find a sort of a new degree of freedom in our ability to interact in specific ways, right, in precise ways with living systems, then you've got an opportunity to be able to address things that seem intractable or seem totally mysterious as diseases in a totally new way. I mean, that's partly, I think, going to have to come from the idea that research is going to have to be taken into space, for example. You really need to push the boundaries of the conditions, the radiative conditions, the whole array of possible processes that are interacting on the system that you're studying. But here, you've got a case where perhaps, as Montagnier points out, there are certain diseases right now which either are hard to identify in terms of their source, or simply hard to treat.
Yes.
That now,
if we could get in there with radio frequencies, what a wonderful breakthrough that would be.
Right.
Yeah, and of course that's this is the man who discovered the AIDS virus, who's been digging into the AIDS virus ever since. He's not convinced that's the whole story. The virus is involved, but he thinks there are other things involved. I think the last time I heard him speaking on this. He thinks the mycoplasma. In fact, one of the reasons he was looking at the mycoplasma. These are very small organisms, mycoplasma, without cell walls. And these pleomorphs, by the way, also one of their features is some of them are get into cell wall deficient forms. These things are very small. These things can get inside cells. These things can hide. If you could, our hope, LaRouche called for a program back when the whole AIDS scare back in the early to mid 80s, when this thing was burgeoning, to mobilize the capabilities, the capabilities of our physics labs, the capability of our national labs. He described it as optical biophysics. The same capabilities which we were looking at from the standpoint of creating a beam defense when LaRouche had made the proposal for a U.S.-Soviet joint effort to develop a beam defense to neutralize the danger of imminent, rapid response nuclear warfare that you couldn't stop. He was proposing, let's mobilize that same capability to an all-out science driver program for use of the electromagnetic spectrum, in the same way we're talking about it now, with a slightly different feature towards NWAPA, but. That was, of course, the first thing that popped into my mind, I think, when I saw this and I saw what Montagnier was doing to develop that capability. And then, of course, it gets into these fascinating broader questions of what we're dealing with. Disease is going to be around a long time. It's there because it's a part of the dynamic process of evolutionary life. It involves this question of symbiogenesis, that things that are adapted, so well adapted to us, and are with us and are in us all the time. The disease process is not simply some invading organism. It has to be some distorted state of the whole totality of an organism in its relationship to a totality of environment. That's where you get into these things that so far we tend to know only statistically, but these questions of why diseases come at certain times, what is their relationship to seasonal cycles and to cycles that must be of electromagnetic radiation that must be working on a solar system and even galactic scale, or extragalactic, where a lot of those cosmic rays come from.
Right. Obviously, there's sort of an idea that there's maybe you start building up, and there's maybe a point at which you get something which is alive. Yet there's there's never been an experiment to prove that. You do have experiments now, recently, Inventor is one of them, which are making certain claims about that get close to saying we're creating something that's alive from something that was non-living before. But yet, as you point out, it always has to be done in the presence of a living organism or something that's alive. It's the ready principle, right?
Yeah, well, that was of course, the paradox. Montagnier mentions what the Venter group had done in the summer of 2010, what they had reported. The paradox in Montagnier's experiment is, well, if you have a test tube over here that had something living in it, although then it went through this multiple dilution process, which also...
And filtration.
And filtration, multiple dilution, and shaking, which is another thing certain people go crazy about. And then over here you had this distilled water. You put it into the solenoid with a 7 hertz frequency, 18 to 24 hours or so, and suddenly this one is now also capable of putting out this signal. But then the final incredible step, you put raw materials, nucleotides, and primers, the things that you would use to do the standard PCR, polymerase chain reaction, except that in the normal case, you would have a sample which serves as what you call the template for the development. So the raw materials are there.
Sample of DNA.
You have a sample of the DNA of the thing you're trying to make assemble into that DNA, and it must, at least as we conceptualize, it must use that sample to get as a sort of template to get the pattern to grow. It appears as I'm understanding what Montagnier is doing, that sample isn't there. That what apparently substituted for that sample was whatever had happened to the water,
right?
Which he wants to term nanostructures within the water, and whatever that radio frequency emission, if that's all it was. I mean, another thing to realize is just because what we're detecting is what appears to be, with relatively simple apparatus, what appears to be a 1,000 hertz signal and then some other things, that doesn't mean that's all that's there. I would be very surprised if that's all that's there.
Right.
I think there's a lot more there. And, I don't even think our conception of what a radio frequency signal is or what light is, these are primitive conceptions because they're conceptions based on simply the physical. But now we see these things are working with life, with a higher form of organization. So what are these things? What is light then? What is radio frequency and so forth? The answer is it's going to be much more interesting when we see how it works on that level. So that raises the question then. Let's just take in that case: was life formed without life? Did we produce life de novo? Well, certainly we had to have we had to have a test tube over here of life to make whatever happened over here. If we weren't fooling ourselves and something else was actually happening, we had to have that.
Not to mention the fact that what was created was a DNA fragment. It wasn't a fully living.
In this case, yeah. In this case, yeah. And then, of course, what the Venter experiment involved, really, the PCR technique can. What Venter demonstrated, I mean, it's not as a technique. It's not to be belittled. It's a marvel, you know. But the problem is there are certain the claims about it and the phraseology in the thing. There's a certain kind of hyping there that's it's only hyping from the standpoint of, I mean, what discourages me about it is simply people don't want to. What has to be challenged is this prevailing reductionist ideology. Mm-hmm. What has to be challenged is the idea that life is only this. Oh, it's easier. You know, you can sell magazines or you know, get a lot of internet hits by saying, "Oh, we created life." So they published a paper in the summer of 2010, creation of a mycoplasma. Was it Pyrum? I forget the species of a particular mycoplasma bacterial cell.
Right.
Well, what they did was they used their techniques, which are marvelous techniques, to assemble nucleotides into this DNA of they used a relatively short. A bacteria that has a relatively simple one. Now, how is the assembly done? The assembly itself involves other organisms. Mm-hmm. You're using other organisms, ultimately a yeast organism, to do the gene combinations. Bacteria do this all the time. You know, bacteria. What we do with genetic engineering, what we've mastered just within the last several decades, is something that bacteria have been doing since the beginning of time, or at least since the beginning of life. So essentially, you swapped out a DNA and put another one in there.
Mm-hmm.
That's not creation of life. It's an interesting thing to do.
Yeah.
Life is something. It's an interesting thing to do, but you know, from the standpoint of technique, I think what Montagnier did, which would be a lesser claim in those terms, in terms of created life. Montagnier is not out there trying to say, I created life.
Mm-hmm.
Montagnier is, I think this is a man committed to understanding the disease process and understanding what is this process of generation.
Right.
The way he's been treated is just horrible. He has to leave his own country and go to what the Chinese are doing for him is wonderful. Of course, they're naming a laboratory in Shanghai, Jiaoteng University, I think. They're giving him, there's going to be a Luc Montagnier Institute there, where he and lots of other people, lots of Chinese students and graduates and so forth will work with him to pursue this work, which is a great thing. It's a sorry thing for Western science, for France and the whole Western science, that anything like that, and it's, to me, reminiscent of cold fusion, that the guys who worked on it were driven out of their own countries.
Right.
You know, people think, oh, don't compare it to that. You know, people don't want to rock the boat. You know, oh, well, everybody who finds something new. You know, I have a lot of experience with this in terms of, you know, editing the 21st Century Science magazine, where we're on the lookout for things that are new and things that don't quite fit, and things that are unusual. You know, and you've always got the question: is it real, or is it? The big question is always: is this real? Is there an outright fraud here? Is somebody just deceived, or is it a real discovery? And sometimes it can even be a combination, not the outright fraud part, but there can be combinations of things. But there's something new there, and always there's this tendency among people because of the environment, that they've got something new, but they don't want to be compared to something else new that might be that's the direction your thinking should be going. Mm-hmm. They're afraid of getting hit, because anybody who gets into something new is hit hard.
Right.
If you've got the credentials of Montagnier and so forth, some people are going to listen to you, and you can go somewhere with it. But we've got to break that. You know, we have to. This mass strike process that is going on, this opening up, has to break out there too, and of course it does. It means freeing the mind from these shackles of you must think this way. And this is why it's a total cultural phenomena. You can't, the political revolutions, if they're not linked to a whole cultural intellectual phenomena like that of advancement, then they go nowhere. Or when they are, then you have a renaissance.